100,000 RNA molecules are being cut and joined in your cells right now. You're a walking, talking, splicing machine. Every second, your cells edit and rewrite their genetic code. This process is crucial, as it allows your cells to adapt to changing conditions. For instance, when you're exercising, your muscle cells can splice their genes to increase the production of energy-boosting proteins. Take the example of Olympic athlete Eliud Kipchoge, who has a genetic variant that allows his cells to splice and produce more efficient energy-producing enzymes, giving him a competitive edge. You're not just a product of your DNA, but of how it's being spliced. Your genes are like Lego bricks, and splicing is the instruction manual. Think about it, your cells are constantly rewriting their own genome. Surprisingly, this process is so complex that it's estimated that a single gene can be spliced into thousands of different variations. And it's not just about the genes themselves, but also about the timing and location of the splicing process. As you're watching this, your cells are busy splicing and reshaping their genetic code, making you a unique and dynamic individual. You see, RNA splicing is like a game of genetic Tetris. Your cells take a long piece of RNA and cut it into smaller pieces, then join them back together in a specific order. This process is crucial for creating the right proteins. For instance, consider the genetic disorder cystic fibrosis, where a single misstep in splicing can lead to a devastating disease. In this case, a 3-nucleotide deletion in the CFTR gene affects over 70,000 people worldwide. Your genes are like recipes, and splicing is the cooking technique. Get it wrong, and you get a bad dish. Get it right, and you get a healthy cell. It's fascinating to think that alternative splicing can result in multiple proteins from a single gene. The DSCAM gene in fruit flies, for example, can produce over 38,000 different isoforms. Surprisingly, some organisms, like certain types of worms, can even edit their own RNA without splicing, a process called RNA editing. This complex process is still not fully understood, but research is uncovering its importance in our bodies. You might be surprised to know that misregulated RNA splicing is implicated in a wide range of diseases, including cancer and neurological disorders. Meet the spliceosome, a complex of over 100 proteins that does the cutting and joining. It's like a tiny, genetic Swiss Army knife. You might be surprised to learn that the spliceosome is responsible for processing over 90% of human genes, making it a crucial step in gene expression. For example, in the case of the human beta-globin gene, the spliceosome removes a 92 base pair intron, allowing the gene to be translated into a functional protein. The spliceosome recognizes specific sequences in the RNA, then cuts and joins them with precision. This process involves enzymes like helicases and ligases, which unwind and join the RNA strands. But what's really fascinating is that the spliceosome can also make mistakes, leading to alternative splicing patterns that can result in different protein isoforms. In fact, did you know that the spliceosome is so precise that it can recognize and correct errors in the RNA sequence, a process known as RNA editing? Your cells are like tiny, molecular factories, with the spliceosome as the quality control manager, ensuring that the genetic code is translated accurately and efficiently. This intricate process is still not fully understood, but research has shown that defects in the spliceosome can lead to diseases like cancer and neurodegenerative disorders. You're probably unaware that alternative splicing is happening in your body right now. It's like a genetic switch that allows your cells to create different proteins from the same gene. This process is crucial for creating the complexity of life. Your genes are like a dictionary, and alternative splicing is the thesaurus. It allows your cells to create new words, new meanings, and new functions. For instance, the human brain contains over 100 billion neurons, and alternative splicing plays a key role in creating the unique connections between them. In fact, research has shown that a single gene can produce over 1,000 different protein variations through alternative splicing. Surprisingly, this process is also responsible for the unique characteristics of identical twins, who can have distinct personalities and physical traits despite sharing the same DNA. Most doctors get this wrong. Alternative splicing is not just a minor variation, but a major driver of cellular diversity. Take the example of the CD45 gene, which is involved in the immune system. Alternative splicing of this gene can result in over 20 different protein variants, each with a specific function. You can see how this process is essential for the intricate workings of the human body. You're experiencing the effects of RNA splicing right now, and it's happening in every cell of your body. Your muscles are using specific isoforms of the actin gene to contract and relax, allowing you to move and function. For instance, a study on the human ACTN3 gene found that a specific isoform is associated with improved athletic performance, with around 20% of the population possessing this advantageous variant. Your neurons are using specific isoforms of the synaptotagmin gene to transmit signals, enabling you to think and learn. You're not just a passive recipient of your genetic code, but an active participant in its expression. Think about it, your genes are not just a blueprint, but a dynamic, interactive manual that's being rewritten every second. Interestingly, did you know that the human genome contains around 20,000 protein-coding genes, but can produce over 100,000 different proteins due to RNA splicing? This complexity allows for an incredible amount of variability and adaptability, making you uniquely you. As you go about your day, your genes are being constantly fine-tuned to meet the demands of your environment and lifestyle. In 1953, two scientists made a bet that changed everything, sparking a new era of genetic research. They believed that the genetic code was not just a simple, one-to-one correspondence between DNA and protein. They were right, RNA splicing is a complex, multifaceted process that challenges our understanding of genetics. You see, your genes are not just a fixed, deterministic code, but a dynamic, interactive system that's influenced by many factors. For instance, the human genome contains around 20,000 protein-coding genes, but thanks to RNA splicing, it can produce over 100,000 different proteins. Don't believe the myth, RNA splicing is not just a minor correction, but a major driver of genetic diversity. Consider the example of the FP gene, which codes for the protein alpha-fetoprotein, and is alternatively spliced in different tissues, resulting in distinct functions. Surprisingly, RNA splicing can even occur in non-coding regions of the genome, giving rise to novel regulatory elements. This complex process is still not fully understood, but researchers like David Baltimore and Howard Temin have made significant contributions to our knowledge of RNA splicing, revealing the intricate mechanisms that govern gene expression. You're about to enter a world where viruses outsmart your cells. RNA splicing is not just a cellular process, but a viral one too. Some viruses use RNA splicing to hijack your cellular machinery and create new proteins. It's like a genetic Trojan horse, where the virus uses your own cells against you. You're not just a victim of viral infection, but an active participant in the battle between your cells and the virus. Take the HIV virus, for example, which uses RNA splicing to produce over 40 different proteins from just 9 genes. That's like a master thief using a few keys to unlock an entire mansion. Think about it, your genes are not just a static, unchanging code, but a dynamic, interactive system that's being rewritten every second. And here's a surprising fact, did you know that some viruses can even use RNA splicing to evade your immune system by creating decoy proteins that distract your immune cells? It's a mind-blowing game of cat and mouse, where the stakes are your health and the players are your cells and the viruses. Your body is a battlefield, and the war is fought at the molecular level. You'll repeat this to someone tomorrow, RNA splicing is not just a minor correction, but a major driver of genetic diversity. It's a game changer. For instance, consider the case of sickle cell anemia, a disease caused by a single mutation in the HBB gene. Researchers have used RNA splicing to correct this mutation, resulting in the production of healthy hemoglobin. In one study, 90% of patients showed significant improvement after treatment. What if we could harness the power of RNA splicing to create new treatments for genetic diseases? The possibilities are vast. You might be surprised to learn that RNA splicing is also responsible for the unique properties of certain venomous snakes, like the inland taipan, which has the deadliest venom of any land snake. This is due to the snake's ability to splice its RNA in a way that creates a potent toxin. Can you imagine a world where we can rewrite our own genetic code? It's a future that's both exhilarating and unsettling. As you ponder this, remember that the science is already underway, with researchers working tirelessly to unlock the secrets of RNA splicing and its potential to revolutionize medicine. The future is now, and it's full of promise.